Hello everyone, I'm Zain.
And I'm Ham.
We are speaking on behalf of our friends Mirna and Jean. Today, we will introduce you to our research entitled A Study of Beta-Thalassemia in a Group of Thalassemic Patients in order to create a genetic database. First of all, we'll provide you with a brief introduction to our research. Beta-thalassemia is a blood disorder that reduces the production of hemoglobin. It affects a large proportion of births. Beta-thalassemia is characterized by reduced or absent beta-globin chains in the HP molecule. Around 7% of the world's population is affected by this disease, which is highly prevalent in tropical and subtropical regions, especially the Mediterranean.
In spite of demographic and ethnic diversity in Syria, there are few studies at molecule level describing mutation of hemoglobin subunit beta HBB gene. There is only one research has reported about geographical distribution of these anomalies in Syria. The aim of this study is to detect the locations of mutations on the HBB gene in order to design a primer for the most mutated location. This way, the mutation scanning cost would be reduced. The genetic detection process for beta-thalassemia disease is done using biotechnology tools as follows.
First, DNA extraction. After collecting the blood samples, DNA is extracted from the white blood cells using QIM DNA blood kit. To isolate and amplify the HBB gene from DNA sample, PCR process is applied on the DNA solution with HBB's reverse and forward primers.
Then, to determine the sequence in the DNA sample, sequencing by synthesis SPS technology is used, which is one of next-generation sequencing NGS methods that generates DNA sequencing data. A solution of one of the four nucleotides required for DNA synthesis, the NTPs, is added to all wells and then washed off. This is done sequentially for all four nucleotides, the ATP, DTTP, TGTP, and then DCTP. If the next base on the template strand is complementary to the added nucleotide, the nucleotide is joined to the growing strand, releasing BBI, which causes a flash of light, is recorded, then the entire process is repeated over and over again. After DNA sequencing, results are generated as FASTA file format and uploaded on Genius Prime. Then, genes of thalassemia patients are compared with a standard gene of a healthy human in order to find the location of mutations in these genes. Therefore, mutations could be visibly detected as shown in this figure. The table shows mutations names and locations on the HBB gene. We notice that most of mutations are positioned between base pair number 45 till base pair number 272.
A study has revealed the presence of many beta-globin gene mutations responsible for beta-thalassemia. These are proportions of mutations spread in the coastal region. The following two, IVS1 110 A instead of G and IVS1 1 A instead of G, are found the most predominant mutations with allele frequency of 37% and 22% respectively. It was found that the rest mutations were less frequent. After the process of detecting mutations, a primer is designed for each one. This way it could be used in PCR tests using these primers to confirm that the patient has a specific mutation or not. However, it's expensive to utilize this technique. Consequently, after the analysis of the outcomes, the observed location of the HBB gene includes the majority of mutations. Thus, a crucial and low-cost step is to design a primer for this high-spot mutated location. Genius Prime is used, and here are the main characteristics of this primer. As we see that the length of target DNA fragment is 410 base pairs, and the length of forward and reverse primers are both 20 base pairs with a binding temperature of approximately 60 Celsius.
Designing a website can play an important role in raising awareness among people and enriching their knowledge about thalassemia, especially those who have it. Our research is uploaded on Thalassemia Syria site, which is also created for the sake of being reachable and helpful as much as possible. This website is designed to be available on all devices. and it has a contact interface which facilitates communication between specialists and patients.
As a result, the blueprints for a genetic database are founded. The mutations that cause beta-thalassemia on the Syrian coast are identified. A high-spot mutation location was observed in the gene sequence. A primer for this spot was designed and came up with satisfying results after testing.
As for mutations, primers could be designed using Genius Prime. Furthermore, the most prevalent mutations would be successfully classified in a genetic database according to their location, frequency, and primer. This technique could be of a low cost and efficient in comparison with other described methods.
To sum it up, detecting the most frequent mutations would help to reduce the screening cost, enhance and facilitate early genetic diagnosis of beta-thalassemia in families and in high-risk couples.
Thus, the illness can be detected at an early point in a previous stage, which can be done by conducting a PCR test, so that a suitable gene therapy would be prescribed for the embryo. Constructing storage units for primers will allow a larger number of people to detect thalassemia earlier, which will help cure them genetically and guarantees the optimum use of primers.
And these are the references of our research.
And thanks for listening.